Hey guys, this is Unit 1, Topic 2, PowerPoint 2, looking at cell differentiation and specialisation. Alright, so I've broken this up into two separate sections in the same PowerPoint. So looking at cell differentiation in the first bit and then specialisation in the second bit. So this part's looking at Pearson 11, Section 4.2. So by the end of the lesson, you need to be able to describe how multicellular organisms have a hierarchical structural organisation of cells, tissues, organs, and systems. Describe how stem cells differ from other cells by being unspecialised and have properties of self-renewal and potency. And describe how stem cells differentiate into specialised cells to form tissues and organs in multicellular organisms. All right, now thinking about unicellular and multicellular organisms, we're really going to be focusing on the multicellular organisms in this section. So we're going to be looking at eukaryotic organisms, more complex and more specialized organisms. All right, so in terms of levels of organization, as we go from smaller, more basic organisms into larger and more complex organisms, there needs to be more coordination between the cells of the body. So we've talked about sort of generalized animal cells and the different organelles and what they do. But we also need to think about the fact that we have a number of different types of specialized cells in the body. So we've got skin cells, we've got nerve cells, cardiac muscle cells. All of those cells have variations which assist complete organs to do a particular job. So in terms of organization, we start off with the specialized cells. From that, a collection of specialized cells make tissues, tissues make organs, and then organs work together to make our different body systems. And obviously those different systems work together in a whole functioning organism to allow life. And this is sort of where we're moving with this unit. We started off looking at individual cells. Now we're looking at whole organisms. And then in Unit 2, we're going to look at the regulation of the whole organism by all of these systems coordinating together. All right, so in terms of specialized cells, I mentioned some examples of this before. Specialized cells are cells in the body that have a particular specialized function. So they perform specific tasks and they have unique adaptations that allow them to do those tasks. So often, like a nerve cell, for example, is very specific in its function and its structure as well. It's not like any other cell in the body. If we take a look at this image over here on the left, this is just looking at some of the different specialized cells and where we would find them. So we've got nerve tissue up here in the brain and nervous system, epithelial tissue, which is for skin. You've got muscle tissue, and we have different types of specialized muscle tissue for our smooth muscles, skeletal muscles, and heart muscles. Connective tissues, so stuff that sort of binds everything together in the body, really important. Blood cells. So all of those things are examples of specialized cells. Now in the textbook, it also goes through some specific examples of specialized cells in plants. So for example, root hairs, the guard cells. They're examples of cells in the plant that do particular jobs. All right, the next level down is looking at tissues. So our specialized cells group together into tissues, and tissues are basically a whole group of similar cells working together to carry out a particular function. The four major types of connective tissue in humans, which you can see in the figure over here on the right, are connective tissue, epithelial tissue, muscle tissue, and nervous tissue. And so basically these are going to make up our different organs. Some examples in plant cells would be the vascular tissue, like the xylem and phloem that we talked about in the last lesson, and dermal tissue, so like the different layers in the leaf, for example, your upper and lower epidermis. Different tissues that do different jobs within the plant cell. All right, the next level is organs, and an organ is a distinct structure made of two or more tissues working together to perform a specialized task. So for example, in humans, you've got the heart, the liver, the brain. In plants, that might be the flower, the leaves, and the roots. So when we say multiple tissues, for example, with our heart, we would have cardiac muscle tissue working alongside nerve tissue, which we need that nerve tissue to be having a signal input from the brain to the heart, as well as connective tissue. So often there's a lot of tissues working together to make these organs work. And finally, systems. So for humans, that could be the circulatory system, the respiratory system, nervous system. We don't have organs in the body that kind of just do their own thing. Typically, the organs in our bodies work together as a system of some sort, and often they are performing the most vital tasks in keeping us alive. You can see that example of the respiratory and cardiac systems working together on the image on the right. And again, with the plant example, it could be the shoot system and the root system. So stuff going on above the ground and under the ground. This is an example from Pearson 11, figure 4.2.4, looking at levels of organisation in animals at the top versus plants at the bottom. Now you can see with our, I'm going to get my little fancy pen, with our human, an example of a specialised cell would be a muscle cell, and that clusters up to make muscle tissue. Now this would be specifically cardiac muscle because it's making up the organ of the heart. The heart is part of the circulatory system, and that's one of the body systems that make up a whole functioning human. Likewise, in a plant cell, the specialized cell might be the root hair, which we talked about in the last lesson, and that combines up to make root tissue. The organ is going to be the roots of the plant, so that's considered the organ of a plant. And the system that is a part of is the vascular bundles, thinking about how we take up nutrients and water and gases through the roots to carry around to the rest of the plant, and that makes up an entire plant. So these levels of organisation are really important because in order to really understand what's going on in a whole organism, we often need to work backwards and look at the systems, organs, tissues, and individual cells and what is important and going on in those cells. Skin is a really, really interesting organ, and it is considered an organ. It's the largest organ in the body and has an average of 1.8 square meters if you were to look at the surface area of the skin. Skin is really important when we look at homeostasis next term because it is the human body's medium between the inside of the body and the external environment. It's what's going to be detecting changes in our environment. There's a lot of sensory receptors, things detecting things like temperature, pain, all of those sorts of things. And so it serves a number of different functions. Things like sweat glands. So you've got sweat glands here. So when we feel hot, we sweat. You've got these little muscles in here that cause your goosebumps, hairs to stand up on your arms when you get cold. You've also got sebaceous glands, which secrete oils. Pain receptors, which are little nerves that are going to detect painful stimuli and tell our brains to move away. So the skin is a really complex and interesting organ that's heavily involved in regulating the body. Okay, so that section was looking at the cell differentiation aspect of this PowerPoint. And in Pearson Biology 11, these are some questions that you could look at on page 134, just to recap before we look at the next section. So the second part of the PowerPoint is cell specialisation. So we looked at these different layers of organisation, but how do our cells go from us as a developing embryo? How do we get heart tissue or cardiac muscle cells? How do we get nerve cells, skin cells? How do they actually specialize into these different types of cells in the body? So section 4.3 in Pearson, we're going to proceed looking at specialization. So we've already covered this first dot point now. Hopefully you can describe how multicellular organisms have a hierarchy of organization going from cells, tissues, organs, and systems. So now we're going to look at that specialization part. So we're going to be looking at stem cells and how they differentiate. Stem cells are a really interesting part of science right now, particularly because of the potential that they have in current medical research and in coming up with cures and solutions for certain ailments. So when we talk about stem cells are cells that haven't been specialized or differentiated into any particular cell types yet. And they have a lot of potential because they can be turned into all of the different sorts of cells that need to make up a human body. Stem cells are basically are the cells that make up a developing embryo. So even though we have some types of stem cells as a living adult, the stem cells that have the most potential are the ones that come from the very early developmental stages of an embryo. Scientists are hoping to use stem cells to replace damaged cells or make cells that our human body might not be making. So in people who have certain deficiencies, they're looking at how we can use stem cells to replace things that should be working properly in the human body. There are a lot of ethical issues around stem cells because if we think about embryonic stem cells and the potential that they have, when we take stem cells from developing embryos, that means that they can no longer develop into a living person. And so there's ethical issues around, is that an ethical thing to do, to take these cells for research and therefore stop a human life from proceeding? All right, so to really understand what's happening with our stem cells, we kind of need to go back to the birds and the bees and talk about what is happening in those very early stages of development. You might recall from year 10 where with fertilization we have the female egg, which has 23 singular chromosomes, and the male sperm, which also has 23 singular chromosomes. They unite during fertilization to make a fertilized egg or a zygote. And a zygote has 46 chromosomes, 23 from each parent. When that zygote implants into the uterus and begins to develop, we call it an embryo. So this is a developing fetus that is going to, after 9 months, become a born baby. While it's in these early stages, we refer to it as an embryo. Now, we kind of have to think to ourselves, we start here as this single cell, a combination of egg and sperm. How does that turn into a baby that has all of these different specialized cells to allow it to live a fully functioning life? It has to have differentiated during this process of development. Now that we understand fertilization and how we get a zygote or a single-celled fertilized egg, we need to look at the early stages of embryonic development to look at where the stem cells are coming from. So if we look down here at the figure, we start with our zygote, and the zygote is a single-celled fertilized egg. In the very early stages after fertilization, the zygote divides by mitosis. So it's just splitting itself and making copies of itself. And after approximately 3 days, we get what's called a morula, which is what's shown here. And that's a bundle of about 16 cells. And that's where we're at before this fertilized egg has even made its way into the mother's uterus to bind. Now, the blastocyst is the really important part here that we're going to focus on a little bit. And this is also shown in the big figure from Pearson on the right here. The morula continues to divide, and it creates this blastocyst, which is like a hollow ball of cells. But there's now going to be a difference between the cells that are on the surface, which we can see here, and what's going on inside the blastocyst. The outside layer are called surface cells, and they're really important because they're what's going to bind the fertilized egg into the mum's uterus. And that's later going to further develop into the placenta, which is going to provide the nourishment for the embryo. The other really important part here is this inner cell mass, the inside of this blastocyst. And that inner cell mass is what's actually going to form our embryo. The rest of it isn't going to be forming the embryo here. Now, we continue to develop into different stages, and this process of gastrulation takes place, which is like a folding of this hollow ball, and we end up with what's called a gastrula. So once it actually implants into the uterus, the gastrula is this structure that has 3 distinct layers of cells, and they're called the endoderm, the ectoderm, and I believe it's mesoderm, which isn't labeled in the figure here, but they are going to differentiate into every single tissue that we end up having as a living human being. We will talk more about this inner cell mass, so just keep that in mind, and there is a video that you can have a look at here in your own time. Now, the layers of the gastrula that I mentioned here, so we've got the, here we go, mesoderm, ectoderm, and endoderm. These layers are called germ layers, and they give rise to all of the different specialized cells that we end up having. The ectoderm is responsible for skin, hair, and the nervous system. Mesoderm forms muscle, cartilage, kidney, and gonads, and the endoderm forms the lungs, bladder, and digestive system. Now, it is from these three layers that our embryonic stem cells are retrieved. So if we just have a look again at the development down here from Pearson, here's our zygote, our morula, our blastocyst, our gastrula, and that's going to develop into the embryo and what we call a fetus after 8 weeks. We can take cells from these germ layers and look at how they differentiate into the specific specialized cells. However, the embryo cannot continue to develop if we interfere with these germ layers. Now, this is showing you a representation of the embryonic stem cells and the potential that they have to differentiate into other cell types. So here we have the embryonic stem cells, and if we break it down into the different germ layers of the mesoderm, endoderm, and ectoderm, these are some of the specialized cells that are going to come from each of those. So again, here's our cardiac muscle, skeletal muscle, kidney cells, red blood cells, smooth muscle cells, all coming from that mesoderm layer. The endoderm is responsible for lung cells, thyroid, pancreatic cells, and the ectoderm is looking at skin, neurons, nerve cells, and pigment cells. So this is a human fetus at about 8 weeks of development. So we can already see some differentiation taking place there. And that's looking at this stage here after the 8-week phase. So what we've talked about so far here are the embryonic stem cells. So they are those undifferentiated cells of the blastocyst, those germ layer cells, which have the potential to become a number of different cell types and continue to replicate into those different cells. The other type of stem cells are the adult stem cells that I talked about, which are present in adults right now. We all have these stem cells. We have some stem cells in the hair follicles. The bulk of our adult stem cells are in the bone marrow. We have some in the spinal cord, some in the germ cells, so they're the cells that are going to differentiate into egg and sperm cells, and they only give rise to particular cell types. So they're more for regenerating damaged cells in our body, but they're not, we can't turn them into other cell types. There's not as much potential for them as the embryonic stem cells have. Now, an example of adult stem cells is shown here on the right with our bone marrow stem cells. You can see that they still have quite a bit of potential, but they're all to do with blood-forming cells. So we can get red blood cells from them. We can get these immune sort of white blood cell types of cells. So they do have some differentiating potential, but we can't turn them into nerve cells or muscle cells. And that brings me to the term potency. And potency refers to the cell's ability to differentiate into other cell types. So going from the most potent to the least potent, totipotent is the most potent cell type. And this gives rise to any cell type as well as a complete living organism. And the only totipotent cells are our fertilized egg and morula. So up here we've got our zygote, and that's because it's a fertilized egg. It has the potential to form any cell type as well as a whole functioning human being. The next level is called pluripotent, and these are the most commonly observed in medical research right now, and they're the ones that we get from our blastocyst layer. They're the ones that give rise to the 3 germ layers that we talked about, and we can end up with a lot of different specialized cells from these pluripotent cells. The next level is called multipotent, and they still have some potential, but this is limited. So this would be our adult stem cells, where our bone marrow, for example, can form multiple different types of cells, but they're all centered around the blood cells, red and white blood cells. Still potential, not as much as our pluripotent stem cells. Lastly, we've got our unipotent cells, which have the least potential, and all that they do is copy the original cell type. They don't differentiate into any other types of cells. So that is things like our skin cells, the cells in our stomach wall, our red blood cells. They don't have potential to turn into anything else other than what they already are. So by the end of the lesson, hopefully in the first part, you looked at the hierarchy of cells, tissues, organs, and systems. And in this second part, you looked at stem cells, how they are unspecialized and can self-renew, as well as have different levels of potency. And then also how stem cells differentiate into these specialized cells to form different tissues and organs. Now there are some questions here from Pearson, and I'm going to scan some of the handouts from Pearson and upload them to the OneNote, which would be the ones that I would recommend that you do, and we can have a look at these on Teams.